Okay, so the point of this video is to get into comparing the different forms of cell division. And it's not going in depth and hitting every name of every single phase within each thing, but just to get the general ideas about how they are the same and different. So when we talk about cell division, where one cell becomes multiple cells, we have mitosis and we have meiosis. Mitosis is all like the MIT. One way to kind of keep these apart is make it twin. MIT, make it twin. So you take one cell and you make identical daughter cells. Meiosis, on the other hand, it has a purpose of producing sex cells, which are variable gametes. So the sex cells are not going to be the same, and we'll look at ways in which variety comes about. When we look at this, again, we'll play with things in clay, but if we just have these clay diagrams here, we've got a cell with an outer cell membrane. We've got a nuclear membrane here, and then we've got DNA on the inside. And I'm skipping the stage where like the spaghetti-like chromatin coils up and becomes chromosomes and sausage-like and condensed and easier to move around instead of being all tangled up with one another. Everything right now is already like in the chromosome form. So we have this original cell, and then you can see here, like we end up with two identical versions of the original. In meiosis, though, we have the original cell, and then we end up with these different varieties here, and none of these are identical to one another, and they are not identical to the parent cell here. They end up with only half as much DNA as the original. So mitosis, again, thinking back to some of the other stuff that we've done here, we begin by duplicating the DNA by replicating it, making a copy of all of the DNA. So this would be happening during the S phase of interphase, but we first need to make a copy of all of the DNA before we can divide it. We get rid of our nucleus, we line chromosomes up in the middle, we pull them apart, we reform the nuclear membranes, and then we split the cell, and we end up with two identical cells to the original. Meiosis, though, looks a little bit more complicated, and there's two sets of division here. So meiosis, again, we start here and we end here, but again, we have this same step where we are replicating the DNA. Then we hook together these tetrads, which we'll look at, and we have some crossing over, but then they line up in the middle, they pull them apart. We have our first division. And then from there, we line them up again, pull them apart, and we have our second division. So let's go a little bit more in depth with mitosis. To say something is diploid means that we have a backup copy of all of the chromosomes, all of the genes. So let's say we've got a section of DNA here from dad and one from mom. Now, a lot of times, both of these um, are going to have genes that are being transcribed. Um, so on this, they would have the same genes, but they may have different alleles for those genes. So for example, you might have a gene that codes for hair color, and you'd also have a gene that codes for hair color on this one. However, this one may be a different version of hair color than on this one. However, the same genes, even though different versions of those genes may exist, the same genes are on both of these. And then we would call these homologous chromosomes, and these would also be a set. So obviously this isn't as complicated as a human cell would be, but for the case of this model, we simplify it to make it easier to understand. But with humans, we'd have 23 pairs. Right now, we just have 2 pairs, but the pair part of this is the diploid part. So the overall purpose of mitosis is for growth or replacement of dead cells. When we say you're making a million new cells every minute, that's mostly mitosis here. So when you start as a single fertilized embryo and develop into trillions of cells, that's a lot of mitosis, of making identical copies of the original cell. Now, you have 200 different kinds of cells, but they all contain the same DNA. So they're still going through mitosis. And you're constantly sloughing off and replacing dead cells, whether they're red blood cells, which have ejected their nucleus and will wear out, or whether they're skin cells, which have all that keratin protein to protect and provide a barrier to everything on the inside, or whether it's liver cells, which are often being replaced as well. But mitosis again, making it twin for growth or replacement of damaged cells. Again, in interphase, we have DNA replication. We are going to create sister chromatids. So again, we have one from mom, one from dad, but then we need to make copies of all of the DNA, because when we end up with two cells, they still need all of the DNA. So during this replication is where we can have mutations, but again, like there's checkpoints in the cell cycle to make sure the fibers are checked correctly, are attached correctly, and things pull apart so you don't have non-disjunction. But with this, we're also having checkpoints to check for mutations and to fix things. And if it can't fix it, then it goes through apoptosis. Again, that's more in the details here. We're just trying to get some of the big ideas. Again, during mitosis, after they duplicate, you lose your nuclear membrane, and then the sister chromatids line up in the middle. The centrioles send out spindle fibers, which attach to either side. You have a checkpoint here where there's a spindle tug of war to make sure everything's attached correctly, and then they pull apart, and then we end up reforming nuclei. So we have two nuclei in one cell, and then ultimately the cell splits in cytokinesis. Now, if we had just continued dividing this, it'd get smaller and smaller and smaller. So then you'd go back into interphase and have a growth phase where things get bigger again. Now, meiosis, again, how do we end up with this variation? We're going to look at that. We're going to look at this two series of divisions. So in meiosis, again, it begins diploid. We contain a backup copy of every chromosome from a parent. Now, we have our replication. So far, this is exactly the same as the interphase and the S phase of interphase that would occur in mitosis. Again, you could have mutations here, which could create some variation. But this point here, we have tetrad formation, which is different than what we see in mitosis. In mitosis, each of these line up individually in the middle. However, in meiosis, in this first part, we have tetrad formation where these homologous chromosomes, because they're coding for the same genes, are hooking up together. So at this point, we have one way that we could have variation is through independent assortment. And when we look at this, right now, we're going to divide these apart, and the dark green would go with the red, and the light green would go with the pink when they divide. It could just as easily have the pink go with the dark green and the red go with the light green. These independently assort from these. So it's not like the light green is always linked to the same side as the pink. These are all independently assorted. When you look at 23 pairs, we could flip-flop all of these things and have a lot of variation in which side is going to be which. Now, here's another form of where variation can occur during this tetrad formation. We can have pieces of chromosomes swapping. So even if this were from dad, a dad chromosome can actually get a piece of mom's. So you can actually mix up the alleles in this way as well. So you can have this crossing over where they split. And when we look at like karyotypes and you start seeing things where one chromosome, they're supposed to be the same length if they're homologous, and one could be like super long, because sometimes this can get mixed up. Instead of perfectly swapping here, things can break off and reattach in different places, and you can get a lot of structural chromosome errors that way. But again, this is typically just adding variation. So we're going to attach the spindle fibers. We're going to pull them apart. Again, some of this stuff is just, it's metaphase, it's anaphase, right? It's very similar to those initial stages where we are pulling apart and dividing the amount of DNA. The first division begins, and then we end up here, and we're going to start beginning the second um, phase of division. So here, again, instead of having one dark green, one light green, and one red and one pink, we end up with variation here. These are not identical to the parent cell. These are not identical daughter cells. So as we start into our second division, again, we have metaphase where things are meeting in the middle, we're pulling them apart, we're getting our second division to occur, and we ultimately end up with four gametes or sex cells. They are haploid, they contain half as much DNA as the original, and none of these are identical to the original parent cell, and none are identical to each other, right? So even with crossing over, even though these are both light green, this could have a little bit of dark green and this could have possibly a little bit of red. And you know how these independently assort, you could have dark green with pink just as easily as you could have dark green with red. Um, but again, this idea that they're haploid, half as much of the total DNA. Now, this idea of meiosis leads us to the idea of Punnett squares. So, when we're thinking about Punnett squares, it's the outer parts of the Punnett squares, which are all of the possible gametes that could be produced from each parent. And like if you have big A, big B, little b, you're going to have an A and a B in each section here. You're not going to have two A's, you're not going to have two greens and then two reds over here. So you're going to have half of each parent, but you're going to contain all of the genes, but not necessarily all of the alleles. So now, again, how do we get more variation again here? So these are different from each parent. We could have independent assortment, which creates differences. We could have crossing over during tetrad formation. But then also, when we talk about the possibility of offspring, this is all random, this fertilization, whether it hooks up this one, or whether it hooks up this one and this one. These are all randomly brought together as far as determining the offspring. So we in these inner boxes are going to be zygotes. These are gametes. These are zygotes. So like homozygous, heterozygous. So these zygotes, these offspring, bring it back to diploid because it was started as four chromosomes. The gametes are two chromosomes. When we bring these back together and fertilize, it's going to come back to four chromosome with a backup copy of each chromosome. So again, this only demos a cell with two pairs of chromosomes. When you start talking about humans having 23 pairs of chromosomes, that's a Punnett square with like 64 trillion possible outcomes here, which is pretty phenomenal. So, quick recap, cell division, we've got mitosis, which is trying to make identical daughter cells, meiosis, which creates sex cells or variable gametes. Mitosis, again, we get identical daughter cells. Meiosis, we get haploid, half as much DNA, and they're all slightly different from one another. Mitosis has one set of division, but again, a lot of the same things are happening. You still replicate the DNA, you still line things up in the middle, you still pull them apart. But meiosis has two sets of division, and we end up with variation due to independent assortment crossing over. But we have to divide things twice. When we go through two divisions, we end up with half as much DNA, and these would be haploid. So those are the big ideas comparing mitosis and meiosis, and hopefully that was helpful.